Moving forward, let's have a look at something different, and I want us to get to the differences between the apes and what we have as the modern man. So, what could possibly be the differences between the apes and modern man? And we will always be able to have this. We have the apes and the modern man. Now, the human beings we have in our environment. And we shall actually be able to say that the ape has a cranium enlarged, and this modern man has a cranium greatly enlarged. So the ape's cranium is enlarged, but the ape's cranium, in terms of its enlargement, is slightly lower than the enlargement of the cranium of the modern man. So the cranium of the modern man is greatly enlarged. And of course, the cranium is just the skull, the bony part of the head region that protects the delicate brain and other structures within the neck and the head region. So the second difference is this: brain capacity is 500 centimeters cubic. So brain capacity or brain size, you can say brain capacity or brain size. And here, the brain capacity stroke size is 1350 centimeters cubic. So this app has a low learning capacity. Why? The cranium is not so enlarged, the brain size is smaller, and of course now there's low learning capacity in relation to the brain size. There's a high intellectual capacity for the modern man. This can get well seen in the way man is making inventions, discoveries, and trying to use the knowledge at his or her disposal to be able to come up with different ideas that can help the world or the environment. The ape has a large incisor and canine. A large incisor and canine used for defense and, of course, killing prey. So the incisor, abbreviated as I in biology, in as far as the rendition of an animal is concerned, and canine, abbreviated as C, in the rendition of an animal or an animal like a human being, we can actually have them being larger. And they are too large or bigger so that they can be able to actually pierce through the flesh of the enemy. So for defense, then they can also kill the prey. You know, there's what we call predation. Predation is a feeding relationship where one animal, the predator, kills and feeds on another animal. The one that gets killed and as well gets fed on is referred to as the prey. So the large incisor and canine of the apes could well get used for defense and killing the prey. Now, here, the incisor and canine are very much reduced. Are very much reduced. Very much reduced. not suitable for defense and killing the prey. Not suitable for defense and killing the prey. Larger incisor and canine, or large incisor and canine, used for defense and killing the prey. Reduced incisors and canines, not suitable for defense and killing the prey. The pelvis for the ape is narrow and elongated. The pelvis is narrow and elongated. But here, the pelvis is broad and as well flattened. The pelvis is broad and flattened. So the hind, the buttocks, to be very specific. If you look at the vertebral column of animals, now the vertebrates, you'll be told that there are 7 bones from the base of the brain to the thoracic cavity. That is to tell you that the first vertebral part of the vertebral column is referred to as the cervical vertebra. The second one is called thoracic vertebra. The third one is called lumbar vertebra. We have what we call the sacral or sacrum. We have what we call the coccyx. You see, or coccygeal. You get the point? So those have to move from the cervical vertebra, from the brain, to the coccyx or coccygeal at the bottom, just at the posterior end of our body. So at the pelvis, at the back of your body, you see, just the behind that you have as a human being, the modern man, is actually flattened and broad so that your sitting apparatus can be able to withstand any pressure of your upper part of the body when you're sitting on a chair, for example. But now that of a monkey is much narrow and elongated, much reduced and elongated. Look at that. So that means the apes that must have existed some years ago must have had a narrow and elongated pelvis, you see, just within your buttock region or your behind. But now that of a modern man is broad and flattened. Flattened to increase the surface area to accommodate the weight of the body when someone is seated, for example, on a chair, and as well be able to support the weight of the body. Now, something else. You'll realize that the pelvis had a quadrupedal locomotion. Quadrupedal locomotion. The layman's language, that means walking on 4. Walking on 4. That means using both the forelimbs and the hind limbs during the walking process. So here the locomotion was bipedal. Bipedal locomotion. Bipedal locomotion, or walking on 2. Walking on 2. Bipedal locomotion. Bipedal locomotion. So look at that. Bipedal locomotion. So pelvis is narrow and elongated. Pelvis is broad and flattened. Quadrupedal locomotion, or walking on 4. Then bipedal locomotion, or walking on 2. So another difference you can talk about, forelimbs. The forelimbs of the ape. Forelimbs of the ape, that is, forelimbs of the ape used for arboreal locomotion. Arboreal locomotion, of course, means maybe jumping from one branch to another, just like the way the monkeys move from one tree branch to another. So arboreal locomotion and of course walking. So you can find a monkey that may use as an app walking on four and using the four limbs for walking. But if found on a tree, these monkeys can jump from one tree branch to another using their four limbs, and as well being able to hold tightly on the branches using their four limbs. So they can use their four limbs for a body locomotion, enabling their movement from one tree branch to another. Four limbs of the apes were used for body locomotion from one tree branch to another, and as well walking on the ground on a flat surface. And of course, they had what you call an opposable thumb. Opposable thumb could help them to get well held on the branch. Opposable thumb, look at that. So they could actually use these limbs to get hold of the branch and use this opposable thumb to tightly hold onto that branch. You see, so this is the branch. They well get held on the branch and use this opposable thumb to strongly hold the branch. So they had an opposable thumb on the forelimbs. Now, forelimbs here, forelimbs are used for manipulation of objects. Manipulation means I can get hold of my calculator, I can open it and be able to begin using it. So I'm using my forelimbs, the hands, to manipulate objects. See, I can use this to open this thing. So modern man uses forelimbs for manipulating objects. For example, modern man can use forelimbs to make tools. So at the end of the day, they still have what you call an opposable thumb. They have an opposable thumb. Have an opposable thumb. So they have an opposable thumb. So opposable thumb. So that can actually give us a good number of differences between the apes and man. And of course, hind foot for this hind foot has opposable toe, that is for the ape. Hind foot has opposable toe for grasping tree branches. And this has non-opposable toe on the hind limbs. Our hind limbs do not have any opposable toe. Our hind limbs do not have an opposable toe. Now the modern man. But now if you look at a monkey, especially the hind limbs have an opposable toe. Not finger again. We're talking about the hind limbs, which can seemingly be able to get used as legs for walking by man, the modern man. So the hind limbs of a monkey have an opposable toe so that it can well grasp the branches. But now the hind limbs of man, the modern man, has no opposable toe. But these hind limbs, now the legs, are mainly used for stability on the ground. And they can actually enable this modern man to walk from one place to another or to actually locomote. So hind limbs in man are used for stability on the ground and be able to move from one place to another. Now that is all about fossil records as an evidence of evolution. And maybe I would want to go to the second evidence of evolution. so that we can be able to really dissect this area and understand how man underwent changes to confirm to us that there was actually evolution. We have what we call geographical distribution. Geographical distribution of organisms. What I would say is this: there's a theory called the continental drift theory that can give us a better explanation. So, geographically, there was distribution of organisms. And the way the organisms became distributed geographically, it can tell us more about how organisms had a similar origin in the first place to begin with. But because of the distribution, these organisms must have undergone some drastic changes. These organisms must have undergone evolution. These organisms must have undergone some structural changes to end up becoming adapted differently to the different climatic conditions in the different areas they had actually destined to after getting distributed. So, what this continental drift theory says is this: there was initially a large, okay, single landmass, a large single landmass. Then this large single landmass underwent drifting. On undergoing drifting, it ended up forming the 7 known continents. So there was a large single landmass that got created. Because of the continental drift theory, the large single landmass underwent breaking up. So it broke up into parts. The parts, after breaking up from each other, drifted from one another, forming the present day continents. We have South America, we have North America, we have the European part of the world, we have what we call the Asia, the Australia, Africa. So those are just examples of the continents we have in our environment. So if you look at these continents, you realize that they must have first initially formed a single large landmass. But because of the continental drift theory, it actually tells us that, or supposes that, these continents were formed because the large single landmass broke up, the parts drifted away from each other, forming those continents. So this continental drift theory isolated the organisms of a common ancestry. For example, if I had the koala bear here, the koala bear and man on the same single large landmass, then the koala bear, due to the continental drift theory separating the parts of this large single landmass, traveled or became distributed or isolated to Australia and ending up becoming a poached mammal. I would end up having man drifting, for example, to Africa to become a placental mammal. So the continental drift theory made it possible for the organisms to become distributed geographically, even if the whole world had organisms with a common ancestry. So those organisms, as they migrated, they evolved along different evolutionary lines. We had a similar origin on the single large landmass. The large landmass has drifted or separated. On separating, the organisms are geographically distributed to the different parts that get formed after the large landmass drifts or separates or breaks up into several parts. On evolving along those different evolutionary lines, it resulted in the formation of new different species, because if we were all mammals here and we migrate due to the drifting and we become distributed to the different continents, we shall evolve along those different evolutionary lines. At the end of the day, what happens? We shall end up resulting into new species, distinct from those found initially in the climatically similar but separate regions. For example, if I decide to actually pick an example of Australia and Africa, these two different parts of the initially single landmass must have had some similarities. They must have been organisms with a particular similarity. But because of the drifting or the separation, this must have ended up making the organisms to move into those two different continents that must have different climatic conditions. At the end of the day, they end up having different characteristics or they actually evolve into new species. Now, you might find some continents having similar climatic conditions, but their flora and fauna, their animals and plants must be different. For example, monkeys with the long prehensile tails inhabit the Amazon forest in South America. So if I decide to actually pick this as my continent, then I talk about the seven continents. I might say this is South America. And then I can call this Africa, I can call this Australia, I can call this maybe North America, then you can maybe use this as South America. Let's pick this. So if I decide to use North America, Australia, South America, and Africa as just examples of continents, not bothering so much about their position, we will realize that monkeys with long prehensile tails inhabit the Amazon forest in South America. Monkeys with long prehensile tails occupy the Amazon forest in South America. But now, look at this. The short-tailed monkeys inhabit the African forests. These monkeys were all having the same origin. They must have been on the same single large landmass. But because of the drifting of the parts of the large landmass to form the present-day continents, those monkeys must have ended up evolving along different evolutionary lines. So they end up finding themselves in different climatic conditions. At the end of the day, they must have evolved to form different organisms or species of the same organisms, like the long prehensile tailed what monkeys in the Amazon forest in South America and the short-tailed monkeys in Africa. Now, another example, the panthers and jaguars found in which category? The class cat are actually found in the Amazon forest in South America. But now, the cat uh, family that you have as leopards and cheetahs are found in Asia. Look at that. So we have panthers and jaguars in the Amazon forest that represent the cat family. We have leopards and cheetahs. Look at that. In Africa. And go to Asia, you'll find the tigers. You see? So it is all about having a particular cat family initially occupying the same single large landmass. It consists of panthers, jaguars, leopards, cheetahs, and tigers. Because of the drifting away of the parts of the single large landmass, the panthers and jaguars have to actually end up getting only found in South America, in the Amazon forest. They are the new species that must have evolved from the cat family during the drifting along that evolutionary line. The leopards and cheetahs found themselves as new species of the cat family in Africa after evolution. The tigers are now found in Asia. From the cat family, because of evolution, they actually underwent changes to become now the cat family representatives now in Asia. Now let's go to this other example. The llamas are found in the Amazon forest as an example of a camel family. We have the modern camels found in Africa. Look at that. So the llamas of the Amazon forest and the modern camels found in Africa and Asia are thought to have had a common ancestry in North America. Look at that. So the llamas and modern camels originated from North America. But because of the drifting, those organisms became geographically distributed. And in the process, they actually underwent evolution. They evolved along those evolutionary lines. At the end of the day, they occupied Asia, Africa, and Amazon forest in South America as areas with different climatic conditions. Because of that, the evolution they underwent made them to become different organisms. Look at that. Now, so the organisms, because of the drifting, became distributed, became isolated, and evolved into different species. Look at that. They became isolated, they became changed, they evolved along those different evolutionary lines, and because of the different climatic conditions in those areas, the organisms ended up becoming different. They evolved into different organisms. So they're not the actual monkeys we had in North America, because you're talking about the llamas in Amazon forest and the modern camels that you know in Africa and Asia. They were the same organisms in North America, but because of evolution, because of the drifting away of the continents, they actually underwent evolution and became different organisms. Now, look at Australia as a continent. It is thought to have been the first continent to drift away from the initial single large landmass. Look at that. It is actually believed that when the Australia drifted away from the initial single large landmass, placental mammals had not actually been formed. Look at that. So this continent ended up having its own peculiar fauna or group of animals. Some of the animals or the mammals found in Australia in the absence of placental mammals, which were not formed when the drifting had actually begun, are like the kangaroos, the koala bears, and the spiny anteaters. So kangaroos, koala bears, and the spiny anteaters are examples of what you call poached mammals or marsupials. They lack a true placenta. Look at that. Kangaroos, koala bears, and spiny anteaters are the poached mammals or the marsupials. They lack a true placenta, like the way the placenta has to actually get so much discussed in terms of the survival of what we call the fetus. So a human being has a true placenta, so is referred to as a placental mammal. But now in Australia, the many examples of mammals you may actually want to give are the kangaroos, the koala bears, and the spiny anteaters. They are actually lacking a true placenta. They are referred to as poached mammals or marsupials. They have a pouch where they can keep their young one. And the young one grows and actually develops to become an adult. Another example of mammals found in Australia is the egg-laying category. Egg-laying mammals or monotremes, such as the dark-billed platypus or echidna, are also found or are very indigenous in Australia. That tells you that, with an exception of human beings, that it is very, very rare for you to find placental mammals, mammals with a true placenta in Australia. Why? Because Australia was the first continent to drift away from the single large landmass before any placental mammals had been formed on that single large landmass. So in drifting away, it only drifted away with one, the marsupials or the poached mammals, and the egg-laying mammals, ending up lacking what you call the placental mammals or mammals with a true placenta. So the drifting away of these continents ended up making organisms to actually evolve along those lines in relation to the climatic conditions of these areas, making each and every continent to have a particular or a peculiar fauna and flora in terms of the animals and plants. Now, you see, as the continents were drifting away from each other, they end up becoming separated by one, deserts, oceans, and mountains. And at the end of the day, this seemingly gave rise to a variety of animal and plant types. Because the types of plants to be found in the desert may not be the same plants you may found on a normal terrestrial what? Piece of land. You see? That's what we call the mesophytes, the hydrophytes, the halophytes, and the xerophytes. like the acacia. So the drifting away from the continents from the single landmass ended up making these continents to become separated by different physical features, such as the oceans, dry deserts, and mountains. And this as well gave rise to different groups of plants and animals. And as far as evolution is concerned, and of course, that's a very nice way of explaining that. And you may also want to actually get more about how evolution occurred. And most importantly, you'd actually want to go to maybe something else so that you can just be able to get a better understanding of this. And what I would want us to actually look at is what you call comparative anatomy. That's the third evidence of evolution. Comparative anatomy. Comparative anatomy. Comparative anatomy. So that is the third evidence of evolution. The third evidence of evolution. Comparative anatomy has well been able to actually maximize on the use of what we call form and structure of different organisms. We look at the structural appearance of the organism. You see, some organisms show basic structural similarities. For example, if I look at a human being and I look at a monkey, these two organisms can show some basic structural similarities. Their body structures can be similar. So if the body structures or the morphology of these organisms is similar, then that can actually trace back into what we call a similar ancestral origin or a common or related ancestry. But where anatomy or morphology is totally different or distanced, then that means they have what we call less evolutionary relationship. Look at that. Look at this. Assuming we have this as a human being and this as a monkey, and they show similar structural features, what does that tell you? They must have a similar or a common ancestry. But if the structures are different or not so much related, then that means those organisms have a different origin or a less evolutionary relationship. That means they must have had a less chance of having been able to evolve from a common ancestral form. So that tells you man and monkeys must have had a similar evolutionary relationship or a common or so related evolutionary relationship. Now, you can find sometimes types of evolution getting discussed here. We have what we call divergent evolution. That means that one basic structural form is getting modified to give rise to various different forms. So we call this divergent. Diverge. Look at that. One, two, three. One basic structural form, like the limb, which can be the forelimb, the hind limb, the wings, maybe in some animals that are actually able to fly, getting modified. One basic structural form, one structure of an organism, or one structure of organisms, getting modified to give rise to various different forms. To give rise to various different forms. Now, when this one basic structure or one basic structural form gets modified to give rise to various forms, then that means at the end of the day that this structure will have to form these forms or these different types of structures now in different organisms, and these structures can actually get adapted to a particular ecological environment or niche. So one basic structure on one basic structural form getting modified to form what actually end up getting that point of forming different structures. These structures can get adapted to different ecological niche in a particular habitat. And this is called adaptive radiation. What is adaptive radiation? where the different forms formed from one basic structure getting adapted, you see, to different ecological niche or niches in different habitats. Now, what are some of these structures? For example, we have what you call homologous structures. Homologous structures. These homologous structures must have had a common embryonic origin, but they are now modified to perform different functions. For example, you might have this as homologous structure. The structure is able to give us different forms. So this can be the first form, the second form, the third, the fourth, and the fifth, and then the sixth. So all these ones are able to get called homologous structures. Why? Homologous structures, I've told you that you can have one basic structural form getting modified. You get the point? As it gets modified, it can form different forms. Those forms can get well adapted to a particular ecological niche or habitat. So adaptive radiation means the different forms resulting from one basic structural form becoming well adapted to a particular or a different ecological niche from the previous form. So if you come here, this can be homologous structures. To mean they must have a similar embryonic or a common embryonic origin, but they have now been modified to perform different functions. So the initial basic structural form underwent modification or underwent evolution to form different basic structural forms. Those forms have actually been adapted to perform different functions. So the structures become homologous structures as long as they have a similar origin and they are now modified to perform different functions. Now look at the forelimb. The forelimb in different vertebrates. You'll realize that the forelimb in the different vertebrates has what we call the basic pentadactyl. Basic pentadactyl. Basic pentadactyl. Basic pentadactyl, or you can also be able to call it the 5-digit. Basic pentadactyl or the 5-digit structure. You see, 1, 2, so you can maybe draw here. We have 1, 2, 3, 4, 5. The basic pentadactyl. 5 digits. The basic pentadactyl or 5-digit structure getting retained. But now, what happens? You'll realize that the functioning is different. For example, if you look at the forelimb of a bat as a mammal, it is actually modified for flying. Look at the forelimb of a human being for grasping. Look at the forelimb of a whale. It is modified into a swimming flipper. And that of even a turtle, the same purpose can actually get captured. If you look at the forelimb of a bird, it's actually modified into a wing for flying. The forelimb of a mole as a mammal, modified for digging. A forelimb of a zebra is elongated and well modified for fast running. So all these forelimbs of all these mammals must have had a common origin with the same basic pentadactyl or 5-digit structure. But because of evolution, because of getting separated into different continents, the structures became more modified. And at the end of the day, they began performing different functions. So such structures are called homologous structures. Homo, similar. That means the structures had the same embryonic origin. They had the same 5-digit or basic pentadactyl structure. But because of undergoing evolution, what happened later? They became modified to perform different functions in these organisms. So the forelimb of a bat modified to form a wing for flight, while that of a horse is elongated to enhance speed in running. The whale or a turtle is modified to have a swimming flipper in as far as its forelimb is concerned. Now, uh, the basic pterodactyl form is modified to perform a particular locomotory function like flying, swimming, walking, and running. Locomotory to mean complete change in position by an organism. Now, another example of an adaptive radiation is that of the beak structures of bats, beaks of bats. And you realize that beaks in bats have a common embryonic origin in terms of the basic structure. And of course, they're just beaks. You can have the hen, the chicken, the dove, the eagle. The basic structure seems to be similar. That confirms that the beak or the beaks of bats must have actually had a common embryonic origin with a basic structure. But depending on the type of food and mode of feeding, what happens? This basic structure has become modified, has undergone evolution, has undergone some drastic changes. You get the point? To suit a particular mode of feeding or the type of food getting eaten by these birds. So the beaks have actually been modified in terms of length, shape, and size. For example, the short, stout, you can actually capture this. The short, stout beaks are common in the seed eaters we have, like the chicken, while the nectar feeders have long, slender beaks to allow their long, slender beaks to find their way into the flowers. Look at that. They can have a slender or smaller in size and long beak so that they can actually find their way into the inner parts of the flower to obtain the nectar. You can also have fruit eaters having relatively large beaks, you see, to actually break or actually separate the parts, you see, or get rid of what you call pericarp of the fruits, you see, so that they can actually have a better meal as far as the fruits are concerned. So the fruit eaters have relatively large beaks so that they can actually be able to have the fruits getting well eaten. Carnivorous birds like the eagles have strong hooked beaks. The strong hooked beaks are able to actually tear the flesh and get hold of the flesh well during the feeding process. So we're just trying to give examples that can actually confirm to us that some structures might have had a common embryonic origin, but because of the evolution, what might have actually happened? These structures might have undergone some changes so that the organisms can actually be able to perform a particular function relating to the habitat, where they're staying, the mode of feeding, and other features to actually get discussed as far as this part is concerned. So we've talked about the forelimbs in mammals, we've talked about the beaks in birds, the beaks in birds. So the beaks in birds can also be able to get captured as far as the way these structures are concerned. Without forgetting this, we'll also be able to actually talk about the foot structure in birds. Just look at the foot structure in birds. The same structure. The same as well pentadactyl structure. But now again, the pentadactyl structure in different birds has undergone modification, evolution, or changes so that each and every foot becomes modified to perform a particular function. For example, you'll actually look at the ducks. They wade in mud. So their webbed feet are able to actually enable them to even swim and wade in mud. If you look at the heavy feet of the eagles, now the carnivorous, they're actually able to grasp and tear the flesh because of their heavy feet. You see? So the heavy feet of eagles, which are actually hooked, can actually get hold of the flesh and tear the flesh as well. So those are just examples of the structures we can be able to actually use in as far as this discussion is concerned. So you might actually want to get more about these structures. Please feel free to be able to have an access to them and be able to see how it goes about. So we can actually be able to actually move to another type of structure. We have structures called analogous structures. So there are those that have different, remember homo means same. So homologous structures have a similar origin, but they are now modified to perform different functions. And in this case, analogous structures are those that have different embryonic origin, but they are now evolved to perform similar functions. Different embryonic origin, but now are able to actually get modified to perform a similar function. With homologous structures, they had the same origin, but now modified to perform different functions. Here, analogous structures, they have what we call different embryonic origin, but now modified to perform a singular or a similar function. So an example, it is actually believed that the wings of insects and the wings of a bat, the mammal, have different embryonic origins. But because of exploiting the same kind of environment, such structures have finally ended up becoming similar in terms of the function they perform. For example, both birds and insects have wings for flight. Recently now, as we say, or as we speak. But the wings of birds have the pentadactyl limb structure, the 5-digit limb structure, while the insect wings are supported by toughened veins, which are composed of cuticle, which originate from the exoskeleton. Remember, we said that in insects, there is a polysaccharide called chitin that actually covers their body, or that actually makes up the exoskeleton of these animals. So if you look at a bird, like a dove, or any bird that flies around, you realize that the forelimb is actually modified into a wing-like structure for attachment of flight muscles. And at the end of the day, this forelimb has still a pentadactyl limb structure, or the 5-digit structure, just modified with a wing-like appearance to allow these birds to fly around. If you look at insects, they are actually able to have wings. The wings are just supported by toughened veins that contain cuticle. And these wings, supported by toughened veins, originate from the exoskeleton. Look at that. At the end of the day, regardless of the differences in the structures, all these structures have been modified because of exploiting the same environment to become useful during flying. Now, look at the eyes of vertebrates. The eyes of vertebrates. I'm a vertebrate. I'm a mammal. I have a backbone. The eyes of vertebrates and those of mollusks, such as the octopus, are also examples of analogous structures. If you look at the structures, they have different embryonic origin. Their structures are literally different, but they have actually been modified to end up performing a similar function in the environment. So analogous structures must have different embryonic origin, but they become modified to perform a similar function. The wings of a bird with a pentadactyl structure are modified to support flying. The wings of an insect with toughened veins, composed of cuticle and originating from the exoskeleton of these insects, like the grasshopper, are also able to end up flying or supporting flying because of the way they have been modified. The eyes of vertebrates like mammals and amphibians and reptiles and the fish and even the birds. And those of the mollusks like the octopus are able to perform a similar function, although they are different in terms of their structures or their appearance. So apart from homologous and analogous structures, we have other structures called vestigial structures. We've talked about homologous. We've talked about analogous. So you have homologous, analogous structures. We have got what we call vestigial structures. So these are structures that have, in the course of time, after a long period of time, ceased to be functional and therefore have become reduced in size or are rudimentary. It was just a large-sized structure initially, but over a long period of time, or in the course of time, has become one, functionless, two, reduced in size, and at the end of the day has become rudimentary with no any signs of growth. Examples: look at the appendix in man or human beings. It does not perform any digestive function. While in herbivores, like the cows, the goats, and the sheep, you see, it is well developed and functions as the part that breaks down cellulose, as far as the alimentary canal of these animals is concerned. I've said the appendix in man has no any function in terms of digestion. It is closely attached to the large intestine. It does not perform any digestive function. But if you go to herbivores, those that feed on vegetation, the appendix breaks down cellulose because these organisms feed on vegetation, and vegetation forms part of what we call kingdom plantae, and plants have cells and the cells have cell wall. The cell wall is made up of cellulose. So then that means the cellulose making up the cell walls of these plant materials or plant cells must be broken down by the appendix during digestion. So the appendix breaks down the cellulose. Number two, we have what you call the python and the whale have no any externally visible limbs. And people still believe that the python as a snake and the whale as a mammal found in the water, as an aquatic mammal, are believed to have had hind limbs. But the hind limbs must have actually ended up reducing in size and actually become rudimentary or functionless. What shows that these organisms must have had hind limbs is because of the pelvic girdle that is still visible. So the pelvic girdle, still visible in these animals, traces back to the presence of hind limbs some time back. Look at the kiwi as a flightless bird of the New Zealand and some other parts of Africa. Has reduced wings beneath the body plumage. It means that this bird under class Fs must have had wings for flying, but the wings must have ended up getting reduced in size over a longer period of time. And after getting reduced in size, the wings became rudimentary. They are still able to actually become visible beneath the body plumage, and they can actually be visible to confirm that they must have actually been larger and functional some time back, but because of evolution or undergoing changes, the wings must have, in the due course, reduced in size, become rudimentary, and now ended up becoming functionless. Look at the vestigial tail in the present human beings. That vestigial tail, we are actually taken to be organisms or animals that must have had a longer tail. But because of evolution, the tail must have reduced in size and become rudimentary or functionless. So that vestigial tail that is much reduced in size is called coccyx. It's called coccyx. Now, look at mammals. Mammals have a nictitating membrane on their eye surfaces. it is reduced and does not perform any function. But if you look at birds and fish, it can move over the eye and act as the third eyelid. So a membrane called the nictitating membrane can move on the surface of the eyes of birds and fish to act as the third eyelid. But now the problem is, in man or mammals, that nictitating membrane has reduced in size and is now functionless. So all these examples of vestigial structures point to the fact that the organisms where the structures are functional and the organisms where the structures are vestigial must have had a common origin. Yeah, that's true. I am a man. I have a reduced nictitating membrane. Someone else or another organism is a bird or a fish, having a nictitating membrane running over the surface of the eye. If we all have nictitating membranes, then that means we must have had a common ancestral what? Origin. But because of evolution, these structures in our bodies underwent drastic changes. And on undergoing these drastic changes, then that means in some the structures became reduced and became functionless or rudimentary, but in some the structures persisted and now can perform different functions. Look at the appendix in man. Look at the appendix in herbivores. We must have had the same origin because we have the same structures, but because of evolution, these structures have undergone changes in such a way that in man, it is now functionless, reduced in size, or rudimentary. But in the herbivores, the structures are still functional, digesting or breaking down cellulose. That's all about comparative anatomy as an evidence of evolution. Another evidence is what you call cell biology. Cell biology. This is another evidence of evolution. Cell biology is another evidence of evolution. So if you look at cell biology, you'll realize that it literally means that you are looking at the organisms, and in that case, you'll first look at their cells. So the cells of all animals show basic similarities in their structure and function. You looked at the cell in Form 1, you are told that there's what you call a plant cell and an animal cell. And what do we say? The cells of both plants and animals contain one, the cell membranes, and other organelles such as ribosomes, Golgi bodies, mitochondria. We have what we call the rough endoplasmic reticulum. So if both plants and animals have similar organelles, what is an organelle? A structure within a cell. Then that means the plants and animals must have had a similar embryonic origin. But because of evolution, we might get some differences between the two cells or the organisms. So one, if cells have the same organelles, like the mitochondrion, the Golgi bodies, the ribosomes, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the cell membranes, then that means they must have had a similar embryonic origin. Now, just to move forward, we have also some biological chemicals, or very important chemicals in both plants and animals. One, we have what we call the adenosine diphosphate, ADP, or adenosine triphosphate, ATP. These are the chemicals that store energy that gets produced in the mitochondrion when food gets broken down chemically in the presence of oxygen during respiration. So ADP and ATP and even DNM, deoxyribonucleic acid, or RNM, ribonucleic acid, all these chemicals are found both in plant cells and animal cells to also tell us that these organisms must have had a similar embryonic origin. Now, or the organisms must have had what we call a common ancestral origin. Now, apart from the similarities displayed in both plants and animal cells, we can also be able to actually look at the differences in the structures and chemical substances in their cells. For example, a plant cell contains cellulose cell wall. You can find a cell wall in an animal cell. So plant cells contain cell walls made of cellulose as a polysaccharide. The cell sap, plants contain a cell sap is a mixture of salt, sugar, and water in the vacuole of plant cells. Remember, a plant cell's vacuole is large and centrally located. But in an animal cell, vacuoles may be absent, but if they are present, they are small structures scattered within the cell of the animal. So, I can find an animal cell, a cell wall. I can find a cell sap, a solution within the vacuole of the plant cell. I can find a chloroplast. I can find starch. You get the point? I can find chlorophyll, the green coloring matter. But all these things are not found in animal cells. To confirm that, even if plants and animals had a similar origin, there must have been evolution. Both plants and animals must have undergone some drastic changes to end up becoming different and having different structures as well. Now, among animals, you can also talk about the blood pigments, the components of blood. For example, we are told that hemoglobin is found in all vertebrates and some invertebrates. Hemoglobin, the red pigment that gives blood its red appearance, the one that combines with oxygen to transport the oxygen around the body, and as well combines with carbon dioxide to transport carbon dioxide from respiring cells to lungs for exhalation. So hemoglobin as a red pigment found in blood is found in vertebrates and in some invertebrates. There's another chemical called hemocyanin. Hemocyanin is also a blood pigment that is widely distributed in both vertebrates and as well invertebrates. But in most cases, it is found in many mollusks, such as the octopus, and some crustaceans, such as the crab, the shrimp, the woodlice, the crayfish, the daphnia. Those are examples of crustaceans that have a blood pigment called hemocyanin. If you go to a phylum called phylum Annelida that has earthworms, you'll find that the chemical present in these members of this phylum is called chlorocruorin. Chlorocruorin. So I've talked about hemoglobin found in vertebrates and invertebrates. Vertebrates such as mammals, birds, fish, amphibians, reptiles, and others. Invertebrates such as the insects and the arachnids. Then if you come to the hemocyanin, it is found in mollusks like the octopus. Then also in crustaceans such as the daphnia, the woodlice, and the crabs and the shrimps. If you talk about chlorocruorin, it is found in members of phylum Annelida, like the earthworms. So, because these organisms have different blood pigment, then that means, yes, they must have had a common or similar embryonic origin. But because of evolution, they must have ended up undergoing some changes to end up becoming different in terms of their blood pigments. So, the presence of the blood pigments or the presence of the blood as a transport fluid confirms that they must have had a common embryonic origin. But ending up having different blood pigments in the transport fluid confirms that there must have been evolution. Very good. So the differences occurring in both plants and animals indicate that from the common ancestral stock, plants and animals became separated and at the end of the day evolved along different lines of evolution. So that's all about cell biology. That's all about cell biology, and we shall also be able to talk about another evidence of evolution. We can talk about comparative embryology. Comparative embryology. So comparative embryology states that you are studying the formation and development of an embryo. Embryology. What is embryology? The study of the formation and development of an embryo. An embryo is a young one of an organism. I remember I was told by my biology teacher that when the male nucleus fuses with the female nucleus, we always form what we call a zygote. The zygote undergoes what we call mitotic cell division. After undergoing mitotic cell division, the zygote forms what we call a mass of cells called morula. The first cell divisions occurring so that you can form a mass of cells called morula is called cleavages. So cleavages is the first cell divisions that occur. When cleavages as first cell divisions occur, they form a mass of cells called morula. This mass of cells can actually travel along the oviduct until they are actually taken to the uterus. Once this mass of cells, forming now the zygote, end up getting implanted in the endometrium or the myometrium wall of the uterus, then that means you're going to have what you call an embryo. So you can only form an embryo when the zygote becomes implanted in the lining of the uterus or in the wall of the uterus. Okay, so the embryos of different animals, like the vertebrates, for example, have been found to have similar morphological or outward appearance features during the early stages of development. Just try to look at the embryos of different vertebrates, like the embryo of a man, the embryo of a dog, the embryo of a cow, the embryo of a lion, the embryo of a zebra, the embryo of a donkey. The embryos or the young ones of this, even the embryo of the chicken, for example, the embryos of these animals have a similar embryonic origin. Why? Morphologically, they are very similar. They have what we call similar morphological features during their early stages of development. In fact, it might be very difficult for differentiate between an embryo of a man and an embryo of a donkey. They are very similar, very similar. That means that if the embryos of these animals are similar, then they must have what we call a similar or a common ancestry. So the closer the resemblance between the embryos in the early stages of development, the closer the phylogenetic relationship in the organisms or of the organisms. So if I compare the embryos of different organisms and realize that they have similar morphological features, they are closer in resemblance, that means they have a common ancestral form, they have a common ancestral origin, they have a similar or closer phylogenetic relationship. Now, this can be defined or explained by what you call recapitulation theory. Let me just try to explain this. It can be defined by what you call recapitulation theory. Recapitulation theory. With recapitulation theory, you will actually understand that the ontogeny recapitulates phylogeny. Ontogeny. I was told this by my biology teacher. Ontogeny recapitulates phylogeny. This means that the stages that an embryo will undergo to form an adult resemble the successive stages in the evolution of the ancestral forms. Let's give this example. This is an embryo undergoing changes to form an adult, for example. So these stages that the embryo undergoes to move from this embryonic stage to this adulthood stage resemble the stages of evolution of the ancestral forms. That's what the recapitulation theory states, of the ancestral forms. So an embryo moves from this embryonic stage to another stage to another stage to another stage until it forms an adult. Those stages resemble the stages of evolution of the ancestral forms. To confirm that, we must have actually had some similarities in terms of the origin of man and whatever has to actually get discussed in terms of evolution. So if my embryonic stage moves to other stages until I form an adult form of the same organism, and those stages are similar to the stages of evolution of the animal, then that means that we must have evolved. We must have undergone some drastic changes. You see? That confirms that we must have undergone some drastic changes in as far as evolution is concerned. We must have undergone some drastic changes in as far as evolution is concerned. So this tells you that if an embryo moves to a particular stage called stage 1, then to another stage 2, to another stage 3, to another stage 3, stage 4, stage 5, stage 6, then that means that those stages are showing how uh, man also evolved from the ancestral ape-like forms to the modern man. And that can actually confirm to us that there must have been evolution, to be precise. If the stages you are actually experiencing right now, in terms of the development of the embryo to the adult form, resemble the stages of what you call the evolution of the ancestral forms to the modern man, that tells us, confirms, that there must have been evolution. 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 So those are the main ways in which you can be able to actually confirm that there was evolution in as far as this topic is concerned. There must have been evolution. Organisms must have evolved. Organisms must have evolved. And there can be a possibility that someone can ask you to also explain evolution in man by using what you call cell serology. Cell serology. So that can also be an evidence of evolution. So we'll actually be able to just give a brief description of how it works, or comparative serology. So that you can be able to understand how it works. So what happens? We have blood, and blood is a very important component of the human body. It helps in one, transporting nutrients and oxygen to different parts of the body, so that each and every cell has an access to oxygen and nutrients for the metabolic purposes. The same blood has to transport wastes such as carbon dioxide and urea as an heterogeneous waste from their site of excretion to where they're supposed to be eliminated from the body of an organism. So blood has blood proteins, and such blood proteins can be albumin, fibrinogen, prothrombin, thrombin, heparin, and others. These blood proteins are able to give us more information about evolution. For example, always remember that antigens and antibodies are mostly discussed as far as evolution is concerned in relation to the proteins found in the blood. We said we have what we call the rhesus factors, antigens and antibodies. We have antigens capital A and capital B to actually give us more information about the blood groups. So it can either be blood group A having an antigen A in capital, or blood group B having capital B as your antigen, or blood group AB having capital A and capital B as your blood group, and as well blood group O with no antigen. Antibodies are written in lowercase, like small letter A and small letter B are the antibodies. So, for example, those organisms or species that have a stronger and common phylogenetic relationship, or those organisms that are phylogenetically related, contain more similar blood proteins. For example, if I'm a man and you are a dog, and we actually have what we call analysis of our blood, and the scientist realizes that the blood proteins in my blood and the blood proteins in your blood as a dog are more in terms of similarity, then that means we have actually ended up confirming that we had a similar ancestral origin. So, similar blood proteins in blood samples of two different organisms tell us that the organisms must have a common ancestral origin, or they are phylogenetically related. Now, experiments are also carried out using what we call a serum. So, experiments with serum are called serological tests. Experiments with serum are also called serological tests. They are actually used to show that relationships exist between different organisms. For example, if the human serum is injected into a rabbit, remember the human serum has both antibodies and antigens. It is getting injected into the body of a rabbit. The rabbit's immune system has antibodies. What happens? The rabbit's immune system produces antibodies to counteract the effect of the antigens in the blood of the human being. In the process, an immunological reaction can occur. Look at that. So the rabbit produces antibodies against the human proteins. When the blood with antibodies is drawn from the rabbit and mixed with the serum from different animals, there must also be an immunological reaction occurring to form a precipitate. So I take my blood, I inject into the body of the rabbit. My blood has blood proteins like antigens. The antigens are able to get seen as invaders or foreign substances. The immune system of the rabbit responds by producing another type of proteins called antibodies. The antibodies fight the antigens from the blood of the human being. An immunological response occurs, a precipitate is formed. Now, if we take blood from the rabbit's body and inject into the bodies of different animals, immunological responses are going to occur to form precipitates. And the amount of precipitate formed varies from one animal to another. And for example, the greater the amount of the precipitate, the closer the relationship between the animal and the human being. I'm assuming I extract blood from my body as a human being, I put in different beakers. Then I extract blood as well from different animals, then add to the blood I've put in the different beakers from this human being. I would expect to find a precipitate forming because of the immunological response, a reaction between the antigens and the antibodies. Now, the reaction between these antibodies and the serum from these animals, such as a chimpanzee, produces a lot of precipitate, showing a close relationship. But now there's what you call a reaction with a distant organism like a dog showing no precipitate. To mean that a dog and a human being are distantly related. They do not have any closer phylogenetic relationship. They might not have had a common ancestral what? Origin. But now a chimpanzee and a monkey with a human being can actually end up having the two blood components from these different animals forming a large amount of precipitate to show a closer phylogenetic relationship or a closer ancestral origin. But any blood from a human being and that of a dog in a particular beaker may not form any precipitate to form a conclusive statement that these animals are distantly related. These animals are distantly related.